I'm Warren Leonard in the Heart, Lung, and Blood Institute, and on behalf of the Cytokine Interest Group, I'm delighted to welcome today's speaker, Chris Garcia from Stanford, who is a world-class structural biologist and protein engineer interested in signaling and making immunotherapeutics. I'm delighted that we're hosting Chris Garcia, a friend with whom I've had the pleasure of collaborating. He is professor of molecular and cellular physiology and the Younger Family Professor of Structural Biology at Stanford, and an investigator at the Howard Hughes Medical Institute. He's interested in cell surface receptors and their ligands, with extraordinary work on cytokines in the T-cell receptor, and using innovative approaches to understand how cell surface receptors engage ligands and transduce signals. He has determined the structure of many cytokine family members, complex with receptors, including common gamma chain cytokines, GP130 family, IL-10, IL-12, and interferons. His lab also determined the first structure for a JAK cytokine receptor complex, the frizzled structure, and much more. He created IL-2 and IL-4 superkines, developed a widely used orthogonal IL-2 system, and has generated a broad range of cytokine partial agonists and surrogate agonists, as well as a SERP alpha variant with enhanced affinity to antagonize CD47. He founded Cytokine, a biotech with a focus on new cytokine-based therapies. Chris received his BS in biochemistry from Tulane and Ph.D. from Johns Hopkins. He was a postdoc at Genentech with David Goodell, and then was then at Scripps with Ian Wilson, and then briefly was on faculty at Scripps before moving to Stanford, where he's been ever since. He's a member of the National Academy of Sciences and National Academy of Medicine, and received the 2021 William Paul Award from the International Cytokine and Interferon Society. First slide, please, and only slide. Finally, I would be remiss if I didn't mention his athletic prowess, including swimming, bicycling, and running with many, many ultramarathons. So it's an absolute pleasure to welcome you, Chris. His title is Immunity Through Ligand-Receptor Engineering.
Well, thanks, Warren. Thanks, Christopher, for organizing this. It's a real honor to give another Walls lecture. I gave one several years ago, and Francis Collins introduced me in the big auditorium, and that was a big thrill. But this is, this will suffice today. I hope to be back in person soon. So, and that picture, yeah, that does bring back memories. That was the Cascade Crest 100-miler in the Cascade Mountains in Washington. Even though I looked like I was pretty happy there, I was very uncomfortable at the time that picture was taken. So, all right, so why don't we get on with the talk here? So just to get a little bit of a background, I think my interest can be summarized as things that happen at the cell surface, mostly ligand-mediated receptor signaling events, and how protein ligands engage their receptors, how they activate signaling, and then using that information, using the actionable value from that information to try and perturb the physiology of the cell. And that's kind of what we're into in here on many different levels and in many different systems. And it involves let me get my laser pointer here, kind of an interplay of structural biology with protein engineering to understand mechanism. And then I use the term actionable value, and that's really my interest in mechanism, is extracting information that we can use for translation to possibly make drugs. And the systems in the lab, to kind of name sort of the highlights here. I've worked in immunity infection for a long time, really go way back with the T-cell receptor. And then, through my experiences at Genentech, I developed an interest in cytokine signaling. So those are kind of the twin poles of the lab, but we also have a big program in regenerative medicine looking at the Wnt and Notch pathways. And then also, most of the receptor systems we look at in the lab are activated by some type of induced proximity. So we're really digging into that now as an engineering platform to induce proximity between proteins on cells that don't normally come together. And I'll talk some about that today. So of all these different kind of projects in the lab, I'm mainly going to focus on cytokines today and really address something that I really got interested in reading Warren Leonard's papers and using this word pleiotropy, which, you know, Warren was really one of the first people to kind of really put this word out there. And it's an apt word because cytokines, they're pleiotropic in that they act on multiple different cell types. Cells have countervailing actions, they have specific actions, and it's a very difficult issue in cytokine biology to really understand what the specific functions of different cytokines are in the face of this pleiotropy. But it's also been a huge barrier to translation, to getting cytokines in the clinic, other than some particular examples, such as EPO and a few others that have very restricted expression of their receptors. Immune cytokines like IL-2 and others have been very difficult to implement in the clinic in an agonist mode. And that's something I've been very interested in based on the mechanism of how cytokine receptors signal. So to the right here, what I'm showing you here is the kind of classic paradigm of how cytokine receptors signal. The cytokine binds with high affinity to one receptor, which then recruits in the second receptor. That causes the Janus kinases, which are associated with the intracellular domains, to transphosphorylate, and then STATs are activated and they go into the nucleus. And this two-step mechanism really affords a lot of engineering possibilities for how we can modify the cytokine or make surrogate agonists to bring these two receptors together in different ways that could modulate signaling. And we've relied a lot on structural biology to provide blueprints of cytokine receptor complexes. This is a selection, an incomplete selection from over the years, starting with IL-1, and you'll see probably one of your favorite molecules up here. We've probably looked at it. And you know, we've really tried to innovate from the structures of the extracellular complexes. And then recently, after sorry, I think I'm going to keep my pointer. Let's see. Well, this how do I get this thing going here? Pointer options. Let's go off there. There we go. Okay. So we recently, last year, a group in my lab, Caleb Glassman, Naotaka Tsutumi, and others, now Nathaniel Cavenet, we determined the structure of a full-length Janus kinase found in the intracellular domain of a cytokine receptor. And I'm going to talk more about that later. But the reason I show this now is really to kind of compare and contrast how we're thinking about cytokine engineering, which is in the context of how GPCRs are activated, and that they're very plastic molecules, and you know, the whole foundation of the GPCR drug discovery industry is this idea that you can make biased agonists and partial agonists. You know, this is a kind of classical pharmacological dose response curve for GPCR ligands that has really been limited to G protein coupled receptors and ion channels, and we're thinking about this kind of engine, this signaling engine, in the context of these pharmacological principles. And that's really driven a lot of what I'm going to show you today, sort of engineering the protein to achieve the same type of behaviors that small molecule agonists for GPCRs work. And also, you know, not just engineering the cytokine, but also making molecules that can change the geometry of the cytokine receptor dimer. So I'm just going to really get started here with a few examples of how we're doing this, and you know, starting off on IL-2, and you know, we determined the structure of this high affinity complex quite a few years ago now, but it's just continued to really yield information that has allowed us to engineer a whole slew of interesting IL-2 variants, and I won't summarize a lot of that, just focusing more on the recent data. And really the interest has been to ask if we can make IL-2 variants that might skew T cells towards regulatory T cells or effector T cells, and so have a lot of other groups, and that's to make drugs for autoimmunity or cancer immunotherapy. And the one recent series of studies we've done is we've made a series of partial agonists for STAT5 signaling by mutating the common gamma chain binding face of IL-2, where we've essentially been able to titrate STAT5 signaling in very fine increments. And we've systematically characterized these in vitro, but also administered them to mice to observe the immune cell profiling behavior, and had some interesting results here. For instance, this one molecule called H9T, which we collaborated with Warren and Nick Restifo on this. It's a very small, it's a partial agonist that's it's very slightly attenuated in its STAT5, but yet Warren took this molecule, and he and his group found some very interesting properties. That it's very IL-2-like in its ability to expand T cells, but there's a reduction in activation of exhaustion markers like TIM3 and LAG, and it also had these stem-like properties as exhibited by the TCF transcription factor, and also its response, its reduced glycolysis, which is sort of hallmarks of stemness. And in working with Nick Restifo, they found that preconditioning cells prior to adoptive transfer in a P-male model significantly delayed tumor growth compared to IL-2 or Super-2, and also led to enhanced persistence. So that was really just the slightest attenuation of STAT5 signaling, really significantly changed the in vivo activity of this molecule, which really shows, how tunable these cytokines are. Here's another example that's a bit more attenuated in its STAT5, which we call REH. And Caleb Glassman in my lab, we also work with Warren on this, found that it has high selectivity for regulatory T cells versus conventional T cells. And we did some RNA-seq with Warren, and found that in TCONs, we saw a reduction in the transcription of certain of these pro-inflammatory molecules, whereas that was less so in the regulatory T cells. So here again, this slight attenuation of the membrane proximal signaling adapter really led to these different changes in different cell subsets of T cells. And we also showed that there was this molecule showed improved treatment of a DSS-induced colitis, intestinal inflammation compared to IL-2, and that is most likely due to this Treg expansion. But we also had kind of an interesting experience with this molecule that, it looked initially to us like a Treg potentiator, but we've also found, since some unpublished data with some collaborators, that it's very effective in tumor models. So this is an MT38 tumor model, and where we know that IL-2, wild-type mouse IL-2 is already pretty effective, but you can't do this in humans. It's just not toxic in mice. And they're Super-2, but this is now this partial agonist, which was CD25 selective, and it expands CD25 high CD8 T cells in the tumor, and it's less toxic than the other forms of IL-2 that we've looked at. So this was quite a surprise and counterintuitive, and really kind of turns this whole idea that in the IL-2 engineering field, that CD25 is the problem. It's referred to as the alpha problem because it's high on Tregs. Well, here we now have a CD25 selective version of IL-2 that really exhibits very profound anti-tumor effects and less toxicity. So alpha may not be the problem. It may actually be an opportunity to selectively target the TILs in the activated TILs in the tumor. Now, moving on to another cytokine here, IL-12. It's another very potent T cell cytokine that has really, been a very tantalizing therapeutic target, except it's extremely toxic, even more so than IL-12. And Caleb Glassman in my lab, a student here, determined the structure of the IL-12 quaternary complex by cryo-EM. And I'm not going to dwell on the structural details here, other than to show you that the interaction of the IL-12 beta 1 with the cytokine is rather unusual in that the green molecule here is IL-12 beta 1, and P40 is the receptor subunit of the cytokine, and it's interacting with the Ig domains of the cytokine rather than the helical bundle, like most cytokines do. And so that led to an interesting opportunity here to ask if we could decouple the activity of IL-12 from NK cells, which secrete a lot of gamma interferon, which is most likely leads to the toxicity of IL-12, and selectively activate T cells. So what Caleb did was he did some staining of activated T cells versus NK cells and found that IL-12 beta 1 is very high on CD8 T cell blasts, much less so on NK cells. So went about with the strategy, if we could weaken the interaction of IL-12 with our beta 1, we might get selectivity for the CD8 T cells. So he made a series of partial agonists by mutating the P40 subunit of IL-12, not the helical subunit. And you can see this 2XL, 3XL, 4XL are now STAT4 partial agonists. And when Caleb looked in a mouse at their ability to activate T cells versus NK cells, you see they largely retained the ability on T cells to make gamma interferon, but the NK cells, now there's much less activity on the NK cells. And so we took this into a tumor model, and this MC38 tumor model again, and found that while wild-type IL-2 is extremely toxic, even at 1 microgram, the mice get very sick until they recover, and then at higher doses, it's lethal. Our partial agonists really had no toxicity whatsoever, but they maintained the same ability to reduce tumor growth and enhance survival. So essentially, we've decoupled the toxicity of IL-12 from its anti-tumor effects. And just in full disclosure, I've started a company called Cytokine that is now putting this into the clinic to test this in humans. Now, changing gears a little bit from let's say cancer immunotherapeutic cytokines to an immunosuppressive IL-10. This is a nice story done by Bobby Saxton in my lab, who's now assistant professor at UC Berkeley. And so IL-10 is a well-known immunosuppressive, especially on monocytes and macrophages, but it also does have some activating effects on T cells, and that's largely due to this. These activities are largely can be separated. The STAT3 is the anti-inflammatory effect downstream of IL-10 receptor, and the STAT1 is the pro-inflammatory. And so Bobby kind of took a similar strategy on IL-10 that I've shown you already. He determined the cryo-EM structure of the IL-10 receptor complex, and it's this interesting dimeric structure. So instead of 2 receptors in one cytokine, you've got a 2 to 2 to 2. And so it sort of departs from the IL-2 type of model that I showed you earlier. But what Bobby did was he found on monocytes that IL-10 beta is high. And so what he did was he created a series of partial agonists on IL-10 to weaken the interaction with IL-10 beta, and then found when he looked at different cell types that he was able to preserve STAT3 induction on monocytes, but significantly diminish it on T cells, B cells, and NK cells, and essentially erase STAT1 signaling from T cells. And so that predicts that we would have preserved the anti-inflammatory effects and eliminated the pro-inflammatory effects. And that was borne out by the gene signatures here. When we did some RNA-seq, these pro-inflammatory cytokines were really significantly reduced with the partial agonist. And we also looked at the actual cytokines themselves, and you can see compared to wild type, the partial agonists are significantly reduced. But in a model, this LPS activation model of monocytes, where they dump out TNF and IL-6 and other pro-inflammatory cytokines, the partial agonist maintains its activity in inhibiting this process, inhibiting the secretion of these cytokines just as well as IL-10. And he also, Bobby went on and did a mouse sepsis model, essentially a cytokine storm model, where he showed that we were able to protect these mice from these IL-6 and TNF cytokine storm when the mice are infected with sepsis as well as wild type IL-10. So here again, we've shown that we can decouple the activities of these cytokines, and getting back to this notion of pleiotropy that I talked about in the beginning. It seems that cytokine engineering is an effective way of addressing that problem. And here's one last piece of data on IL-10 in a colitis model. IL-10's had a lot of interest in the clinic for inflammatory bowel disease, but there's been this problem of this pro-inflammatory aspects of wild type IL-10. And here we show that we can protect mice from DSS as well as the wild type cytokine here. And this is also on a clinical path through Cytokine. All right, and so finally, I want to finish up here on gamma interferon, which, arguably next to IL-2 is the premier immune master regulator and is even more pleiotropic than IL-2. It's produced by many types of cells, and it acts on most types of immune cells. And so when we started, there was not a structure known of this receptor complex, and Juan Mendoza in my lab, who's now a junior faculty at University of Chicago, took on this challenge. And really the big vision we had for this, the rationale for getting into this would be to ask, could we uncouple the immunostimulatory from the immunosuppressive actions of gamma interferon? I mean, the principal action of gamma interferon, which most people know about, is that it upregulates class 1 expression, but it also upregulates checkpoint ligands. So it's immunosuppressive in the tumor microenvironment. So we wanted to ask by structure if we could possibly decouple these actions. And so Juan determined the structure of the gamma interferon receptor complex. And you can see here, gamma interferon bears some resemblance to IL-10. It's a dimer, and it's decorated by these 4 receptors, 2 gamma R1s, 2 gamma R2s. And I'm not going to get into the details of this one either, but just to show you what Juan did here, he selectively ablated the different cytokine binding sites, receptor binding sites on gamma interferon, and in so doing created a series of partial agonists. This is PSTAT1 we're looking at here. And this was a beautiful calibration of the Emax range of gamma interferon. And so with these partial agonists then, we went and did some RNA-seq experiments, look at gene expression, and found something rather unexpected, which is that most of the genes upregulated by gamma interferon were upregulated to roughly the same extent by wild type gamma versus the partial agonists. And we call those stable genes. But there was a subset, a much smaller subset, where the partial agonists upregulated genes to a much lower degree than the wild type. And amongst the stable ones was class 1 MHC, and amongst the tunable ones was PDL1, the checkpoint ligand. So with that information, Juan and Leon Su in my lab, and collaborating with Ed Engelman, we started staining cancer cell lines. And here's an example of lung cancer cell line where the upregulation of HLA-A, this class 1, is equivalent between wild type and the partial agonists, whereas the PDL1 is attenuated significantly by the partial agonists. And Juan went on to do this experiment with many different types of cancer cell lines. And there's bias to different degrees, some more, some less, some almost non-existent. But we do seem to be able to tilt the balance towards class 1 over PDL1 in generally. And so what we're doing now in my lab, Xiang Zhao in my lab has now made some mouse gamma interferon partial agonists that finally allow us to explore the properties of these biased molecules in vivo tumor models. So that's where that's going. So just to summarize what I've shown you, what have we learned? That cytokine, that cells have intrinsic response thresholds to cytokines that can be exploited to decouple cytokine pleiotropy. And those response thresholds are governed by receptor expression, by STAT activation levels, and by gene expression. It's a multi-level control of these hardwired response thresholds. But the bottom line is that by making partial agonists of cytokine, we can, in many cases, phase out actions on different cell types and significantly narrow the pleiotropy. In this case, for example, Bobby Saxton was able to narrow the activity of IL-10 to essentially only monocytes. And so really, I think the path forward for getting cytokines into the clinic, is probably going to incorporate this concept here of tuning down the agonism and getting better cell subset selectivity. All right, so until now, what I've shown you is engineering the cytokine itself, sort of tuning it down, lowering its activity. But there are a lot of other ways of doing this that if you get away from the cytokine. I mean, the cytokine, and I'm showing you here to the right, this is a 4-helix bundle classical model system, EPO bound to the 2 EPO receptors. And also here we now have a GPCR, and I'm showing you the kind of conformational changes that GPCRs undergo with the G protein in response to different ligands. And we took another approach here, which is to ask, instead of just down-tuning the binding of the second receptor, could we reorient the dimer geometry to accomplish something analogous to the shape changes that GPCRs undergo when they bind their ligands? But to do that, the cytokine scaffold itself was not suitable. That's just not, there's not a lot of opportunities to reorient the dimer geometry here with that scaffold. So what we did, so what we'd like to do is we'd like to have a scaffold that we can change the dimer geometry, to change the proximity and orientation of the JAKs to one another, to tune the signaling output, much in the way the medicinal chemistry is able to make small molecules that can change the shape change of GPCRs. So that was the overall concept. And we went about this by using a different binding scaffold, which is called a DARPin. And the DARPin is essentially a professional binding scaffold. By that I mean a synthetic binding scaffold that was created for the sole purpose of selecting binders from libraries. And so Kritika Mohan in my lab made a DARPin library and selected against the extracellular domain of EPO receptor. And this is the crystal structure of that DARPin complex with EPO receptor. And then we collaborated with David Baker's lab to see if we could dimerize the DARPins and bring in a second receptor to maybe activate signaling. And we were able to do that. We designed with the Baker lab these mutations in the dimer interface that compelled the dimerization of the DARPin, and that brought 2 receptors together. And that has been a fantastically useful tool because the DARPins are very engineerable. You can insert these little helical repeats into DARPins and change their curvature. And we did so. We made a series of different angled DARPins, and you can see how they're changing the angle of the EPO receptor dimer here. And at the same time, Kritika measured STAT5 and ERK signaling. And you can see as we increase the dimer angle, we start to phase out signaling. So as the receptors start to lie flat on the membrane, the JAKs just seem to be unable to communicate with each other anymore, and we don't get activation of the receptor. And then there's another way of inserting the helical repeats into the DARPins that gives you a change in the translational, in the distance between the monomers rather than the curvature. And so that just enables a pure separation of the receptors, which you can measure. And in that series, rather than a gradual phasing down of the Emax. We got pretty much maintained with increasing distance the Emax of signaling through STAT5, and then at some threshold distance, it broke off. And clearly the JAKs at that point were not able to talk to each other. So we have two different kind of receptor tuning here through this is a geometry and this is distance. And we went on to ask about the physiological relevance of this kind of approach, working with Vijay Sankaran at the Broad, who had this stem cell differentiation assay from hematopoietic stem cells to red blood cells, where you can conveniently follow CD31 and CD235, and they make this stereotypical U-shaped curve over the course of development. And this is showing you what EPO looks like after the 14th day or so. Well, we found that by using these DARPins that freeze the receptor in different angular geometries, we can essentially abort red blood cell development at different stages, from essentially early stage to the more upright angles, the more later stages, which phenocopies EPO. So this is an example where just simply changing the receptor orientation really had profound effects on cell fate determinations. And that's something that I remain very interested in, but in doing it in a slightly different way. So the DARPins are interesting, but they don't have drug-like properties. They're kind of a synthetic tool compound, which you can do very interesting protein engineering things with them, but the step towards putting them into humans in systemic circulation is really not, it's not been proven yet that that's possible. So we wanted to achieve what I just showed you, which is this geometric reorientation of the receptor extracellular domains, but not using a DARPin scaffold. So for this, we turn to a nanobody dimer scaffold, either two nanobodies linked together like beads on a string, or a nanobody or an scFv, essentially small antibody binding domains that would hopefully dimerize two receptors here, like in this case, IL-2 brings together its two receptors. And so this was work done in my lab by Michelle Yin and Kevin Jude and multiple people in my lab were involved in this project. And so what we did was we collaborated with Yakun Wan in China to make nanobodies against the extracellular domains of IL-2R beta and common gamma chain, with the logic being that these nanobodies will bind to different epitopes on the ECDs. And so that when we link them together, they will bring the two receptors together in different geometries by virtue of the fact that they're binding to different parts of the receptor extracellular domain. And we can make a matrix then of these nanobodies, the beta binders versus the gamma binders, in two different orientations, a forward and a reverse orientation. And in that way, we get this matrix of geometries that we can very easily test on molecules, on cells for STAT5 activation. And when Michelle did this, she found a real diversity of activation strengths of the different binders, from weak activators to activators that had the same Emax as IL-2, to even some of them super agonists that were even stronger than IL-2. And this wasn't just an activation strength issue. If we see bias too, so when Michelle looked at phosphostat5 and phospho-AKT, you can see that as we look at different versions of these molecules, we can see different degrees of bias of STAT5 over AKT. And when Michelle looked at the function, did some functional assays with these molecules, for example, looking at CD8 T cell differentiation from PBMCs, she found that different surrogate agonists could be binned into effector memory biased, effector central memory biased, or central memory biased. And this just really was just a really fascinating result, I thought, because this is due in large part to simply the fact that compared to IL-2, here's the native IL-2 receptor geometry, these surrogate agonists are dimerizing the receptors in different shapes and different distances between the intracellular domains in very unpredictable ways that are having these kind of effects on signaling and function. So essentially, this is a way of taking a single cytokine and then creating a huge number of different flavors of that cytokine that have different functions. And so we're doing this in a lot of different systems now. Each, variant might have a different place in immune disease, a different therapeutic indication. We've also done this with type 1 interferon agonists, where, again, we made nanobodies against the extracellular domains of IFNAR1 and IFNAR2, and then did a matrix all against all. And here we found a much lower hit rate of agonists. So interferon receptor seems to be much more structurally restrictive in its orientation than IL-2 receptor. But nevertheless, we found some hits. And just showing you here now, they activate, they're partial agonists compared to type 1 interferon and A549 cells. But even though they're partial agonists, they're just as potent as type 1 interferon, for example, in inhibiting SARS-CoV-2 replication in A549 cells. These are dose response curves for inhibiting SARS-CoV-2 replication. And in working with the Barrick and Diamond labs, we've also found that they inhibit other types of viruses like Zika virus, in fact, more potently than type 1 interferon. But the real part of these molecules that I find interesting is that, one of the limitations of using type 1 interferons in the clinic is that they have a lot of side effects called flu-like side effects. They activate a lot of pro-inflammatory genes. And our surrogate agonists barely activate these pro-inflammatory cytokines here compared to type 1 interferon. You see they're greatly attenuated. So our prediction is they're going to have much less side effects as type 1 interferons. And I've started a company, Topspin, that is going to try and take these into people as a kind of next-gen antivirals with less side effects. So I want to go from here. So until now, we've been talking about known cytokine signaling pathways, tuning known systems with different kinds of molecules. We're also thinking about this in a different kind of way, which is bringing together cytokine receptors that don't normally pair on the cell surface through induced proximity. And if you think about it, all the pleiotropy that Warren has written about and published on so prolifically is also an opportunity to create new cytokine signaling axes that don't exist in nature. Because every type of immune cell has multiple different kinds of cytokine receptors. And so you can sort of think of that as like a menu of possible ways of bringing them together. And when you think of that, we have something like 30 or 40 different cytokine receptors, and that the kinases associated with the intracellular domains have the ability to crosstalk with one another, it really opens up a whole new possibility of creating new cytokine signaling axes. So here's one example that was principally done by Junming Ren in my lab, where we took the IL-2 receptor complex and the IL-10 receptor complex, and we borrowed a chain from each complex to try and make an IL-2R beta, IL-10R beta heterodimer using this nanobody type of approach. And Junming found a number of hits that activated STAT5. We don't see STAT3 activation. So this is now just activating STAT5. And in some early functional assays of this, he finds that, different versions are able to activate CD8 T cell proliferation to different degrees. They're also able to activate NK cells, although not as potently as IL-2. So what we really have here is like a new flavor of IL-2, because we've replaced common gamma chain with IL-10R beta. That's going to have its own unique constellation of signaling adapters that we're in the process of exploring. But so this is a cytokine that doesn't exist in nature, but has activity on natural cells. And so we're looking into this a lot deeper and really trying to understand what new properties these synthetic cytokines have. So I want to pivot a little bit here from systemic cytokines, which our lab is mainly a protein lab, and so we're more interested in making protein molecules that do things on cells. But cell therapy is also a great opportunity for protein engineering, and I'm going to show you what our contribution to advancing cell therapy has been. And that's been this orthogonal IL-2 system. So what I'm showing you here is the IL-2 receptor complex again. And so, one of the limitations of cell therapy is that when you give people engineered T cells, they don't expand as much as you'd like them to, and you'd like to give them IL-2 to expand those cells, but IL-2 is very toxic. So we thought if we could make an IL-2 that was selective for IL-2R beta, that would be one possible way of solving that problem. So what we did was we made an IL-2, a mutant IL-2 that no longer bound to wild type IL-2R beta, and a mutant IL-2R beta that no longer bound to wild type IL-2, but that these mutants bound to each other. And the idea being that you can then install that mutant receptor into engineered T cells, and then put those cells into people or mice, dose with the mutant cytokine, and get very selective expansion of the engineered T cells. And in fact, it's worked beautifully. And these are the different kind of things you can do with it. You can expand cells. We can transplant without host preconditioning. There's reduced toxicity, mitigating exhaustion. There are a lot of reasons why that this is a very powerful technology. And just to show you a little bit about how we did it, this is the original engineering was done by Jonathan Sokolowski in my lab. And the way he went about it was making a mutation in the receptor so it abrogated IL-2 binding. And this is done based on examining of the structure, and then creating a library on IL-2, and then selecting against the mutant, selecting for the mutant and against the wild type receptor. So here's what this looks like. Basically, this is now the library binding to IL-2, the mutant receptor that doesn't bind to IL-2 and wild type. And you can see neither of them are really binding very well. But after the selection, you get selectivity for the mutant receptor versus the wild type receptor. So now we have what we call an orthogonal IL-2. And what this looks like is that when you transduce T cells with the ortho beta receptor and then add the ortho cytokine, you get a STAT5 signal. Whereas when you apply the orthogonal cytokine to wild type T cells, we don't get any cell growth in this case. And we've demonstrated this in vivo. Jonathan and also Leon Su in my lab have done experiments where we can mix the ortho2 cells with normal T cells, and we get very specific expansion of the ortho T cells by the ortho cytokine. And that is not, we do not see expansion of cells that do not have the orthogonal receptor. And we've shown with Mike Malone here, here's one example in a CAR T model, in a CD19 CAR T, where we put the orthogonal receptor into the CAR T cells, and then you can see dose-dependent expansion of the T cells at different doses of the orthogonal cytokine. This is log scale. So this is 2 log expansion of these cells. And in this experiment, Mike showed very nicely that the CAR T without the orthogonal system is ineffective at controlling these tumors. But when you add the orthogonal cytokine, you get complete tumor control. And the nice thing is that you can dose significantly fewer cells that have the orthogonal. For example, this is 200,000 cells. It's a little bit effective. And this is 500,000 cells, which is just as effective as 1 million cells. So it allows you to really put in many fewer cells into the mice. And also the survival benefit is clear. And this is also another molecule which is going into the clinic through Cytokine. Now, there's been a recent twist with this ortho beta system that I want to tell you about. And this was a collaboration between my lab, Tony Ribas, Carl June, and Anusha Kalbasi, who's now here at Stanford. And what this system does, it affords you the possibility of swapping out the intracellular domain of the beta receptor for any of the common gamma cytokine family. And here's a picture from one of Warren's reviews on all the common gamma chain cytokines. So this inspired us to replace the intracellular domain of beta with each of the common gamma chain cytokine family members. And this was a really interesting experiment. So this is now showing you all the different ICDs on STAT5, activating STAT5, STAT3, STAT1, and STAT6. So predictably, the ortho4 receptor only activates STAT6, but ortho9 activates 1, 3, and 5. And so it's kind of the Swiss Army knife cytokine receptor activating almost all the STATs except STAT6. And the other ones are interesting too. I mean, ortho7 is principally STAT5 focused. It's kind of a partial agonist for STAT5. But we published this paper last year with Anusha as the first author, working with Leon, Sue, and Miko and Carl's lab. And I'm just going to focus on the ortho9 for now, because what we also found was that if we look at, effector markers like CD62 and stemness markers like FAS and SCA, ortho9 and ortho21R seem to have advantages over the other common gamma family members. I mean, especially when it comes to stemness. So we chose ortho9 receptor to go a little bit deeper into in vivo studies. And this is some of Anusha's work. And he showed that ortho9 is able to enable tumor clearance in adopted cell therapy in the P-male model in the absence of lymphodepletion. And not only that, but you see better infiltration of the engineered T cells into the tumor, showing here's the Tisney plot of ortho9 compared to ortho2. The ortho9 cells have more of an activated phenotype than the ortho2 cells. And they're also the stemness markers. This is ortho9 right here. The stemness markers are up and reduce exhaustion markers and things like that. So this was really surprising. I mean, IL-9, really, we don't know a lot about IL-9 or IL-9 receptor signaling, what it's really doing. There aren't many types of T cells that are responsive to IL-9. But when we take that intracellular domain and put it into a completely new context, it reveals these functions that we really had no idea about. And this is some data from Carl's lab, which supported Anusha's data in this chimeric, this CAR T for solid tumor, this mesothelin model. And you can see in an incucyte experiment, here are these CAR Ts that are supported by ortho2, and here they are with ortho9. And so there's much better cytotoxicity by the ortho9. There are also enhanced effector functions and also the stemness and this is now in a solid tumor model. So really, the ortho9 system is super interesting, and we're looking deeper into it as perhaps the next gen beyond ortho2. But there's one liability of the ortho9 system compared to ortho2, which is that ortho2, the cells expand tremendously in the systemic circulation. Ortho9 does not have a strong proliferative signal. So what Leon Su has been doing in my lab is he's taking a hard look at the IL-9 receptor intracellular domain tyrosines, and he's been mutating them and doing some protein engineering on the intracellular domain. And you can see now when we look at the STAT5 signaling, STAT5, STAT1, and STAT3, really don't focus in on the details here. Just look at here's ortho2 versus ortho9 wild type, and you can see it looks like, as we showed earlier, ortho9 activates all of them. But Leon has made versions which now can tune the degree of stat activation of the different stats. And at the same time, testing these in proliferation assays, he's now been able to essentially rescue the proliferative activity of ortho9 here in this mutant. It's not as good as IL-2, but it's still something, and while also preserving its stemness properties to different degrees. And so we're beginning to work with Mike Malone and Anusha on testing these new variants in the same tumor models to ask questions about, what is really the best combination of signals here, from these tuned mutants here. But it also just really highlights again this incredible plasticity of cytokine signaling that just really affords the opportunity of tunability for therapeutics. So I'll kind of finish up the cytokine engineering section a little bit here, and really, all these cell fate decisions are really made in the context of a cytokine milieu, and we think that the kind of experiments I've shown you so far really open up a lot of possibility of tuning cytokines early in these fate determination pathways, and possibly creating new cell fates or altering fate choices. In addition to the peripheral differentiated cell types that I've talked about so far. So that's kind of the direction we're going. Now I want to finish up here, really getting back to mechanism. After a long, I started working on the JAK structure when I was a postdoc at Scripps, and finally in 2022, through cryo-EM, a team of people in my lab, Caleb, Naotaka, Kevin, recently Nathaniel, we managed to determine a structure of a full-length JAK, and so in this structure, in the one on the left here, the kinase domains here were apart, and we know that when the JAK, so that this is the intracellular domain, when it binds and the cytokine brings these together, these kinase domains have to transphosphorylate, in other words, to activate further signaling. And so recently, Nathaniel and Bobby Saxton in my lab and Deepa Wagre managed to trap a conformation of the kinase domains where they're in communication with each other. So now this is the active transphosphorylating pose of this one. And so the message here, there's a lot of flexibility in these kinase domains, and you would imagine there has to be, because these intracellular domain tails kind of hang off here and kind of wind their way down to the kinase domains, and the kinase domains have to be able to access different tyrosines on these intracellular domains, so they need to be somewhat mobile. And so we're continuing to escalate our structural studies here to now trap a STAT complex with the JAK. But what I want to also show you today is what we learned about oncogenic mutations from the structure. So there's this classic mutation in JAKs called V617F, which is responsible for many myeloproliferative neoplasms. It's a constitutive activating mutation in the pseudokinase domain. And this is a figure from the original paper. Apologies, I don't have the reference here. But this was several groups determined this at the same time about 15 years ago. And this mutation maps right into this dimer interface right there between the pseudokinase domains. And it's a valine to a phenylalanine. And so what it does, if you look at the wild type interface between the pseudokinase domains, it's a very poorly packed interface. And so in this model, the cytokine is really responsible for bringing the two receptors with their JAKs together to form this dimer. However, when the valine is mutated to a phenylalanine, like in V617F, it fills the gaps and it improves the shape complementarity between these domains. And when that happens now, the JAKs are able to dimerize without the requirement of the cytokine bringing the two receptors together. So it's essentially an on-mechanism mutation that just serves to cement this dimer together in a cytokine-independent way. And we did some experiments with Jakob Pieler using a single molecule turf several years ago, and Jakob and his group showed that this constitutive homodimerization in the absence of ligand is how V617F works. So for instance, here's TPO receptor and EPO receptor, and this co-locomotion is just another word for dimerization on the cell surface. And you can see when the wild type JAK is mutated to V617F, both receptors now dimerize in a ligand independent fashion. So now with the structure, we're able to kind of put all this together, and this ligand independent dimerization is due to this increased shape complementarity as a result of this phenylalanine mutation, which forms kind of a glue between the JAKs to hold them together. And there's all kinds of other mutations in JAKs outside of this V617F that are also activating. that don't work through dimerization. And this is another figure from Jakob's paper, where here we have these constitutive activating JAK mutations. They don't cause constitutive dimerization. And so we're really interested in this. And, these JAK mutants, John O'Shea, there at NIH, and congratulations to John, he was just elected to the National Academy. John really pioneered the development of these JAK inhibitors to inhibit these kinase domains right here and treat myeloproliferative neoplasms. Well, that's now like a standard of care in the clinic now. It's a pillar of therapy. We're thinking about other ways to do this. And one way is through recruiting phosphatases to the receptor. So in this case, the V617F would be signaling constitutively. If we can bring a phosphatase to the receptor and then have the intracellular domain of the phosphatase directly dephosphorylate the receptor instead of direct inhibition of the kinase, we might get more specificity, because we can have you do this with a bispecific antibody specific to the specific receptor. And so that's kind of one thing we're working on that I'm going to leave you with. And in some preliminary data, this, what we call Ripper molecule, is able to inhibit proliferation of cells bearing this JAK2VF mutation, almost effectively as good as the JAK inhibitors. And so this is kind of a proof of concept experiment that we can dephosphorylate that JAK. And we're sort of, this is just the beginning here of trying to explore this as a new way of thinking about inhibiting oncogenic receptors. So I'm going to end here and just tell you that we're 20 years later, my lab is more excited about cytokine receptors than ever. This is kind of our menu of candidates here. When you think about the diversity of the extracellular domains, their tissue expression, how the JAKs talk to each other, and putting that together with the kind of technologies I've talked about today, I sort of think about cytokine receptors as like an artist's palette of colors that we can mix and match in different combinations and create new shades and new hues. And so we're having a lot of fun with that now, and hopefully we'll have some new things to tell you about soon. So I want to finish by acknowledging all the people who did this work. I tried to mention names along the way, so I don't really need to go into detail on this slide. You've probably seen the names mentioned here or their publications. So I want to thank all these people for their contributions over the years, and also again, Cytokine is commercializing most of the molecules that I show today. So I'll stop here and happy to take any questions.
Okay, Chris, thank you very much for that wonderful talk. Let me remind people to send live feedback with questions so that we can see them in the chat. Let me start with one question, Chris. Besides IL-2 and making an ortho IL-2 molecule, do you have a sense that other ortho cytokines would be of value to create something of a parallel system where you would put in a different cytokine receptor in and have a different paired cytokine?
Yeah, so non-overlapping ortho cytokines, so you could selectively activate two different pathways.
Yep.
Yeah, no, absolutely. I mean, I think orthogonality, I think people are starting to finally wake up to orthogonality and all the sort of therapeutic implications it has. Absolutely. And orthogonality in practice, I made it sound easy, but it's very difficult to achieve, to find a protein that will have absolutely no reactivity to any existing protein in the body. So it's non-trivial to make orthogonal interactions, but if you can, it's very powerful. So what we're also doing now, I have a new postdoc, Ping Dong, in my lab. He's trying to make the double ortho. In other words, not just ortho against beta, but ortho against gamma. So instead of being limited to one ICD change, we can now explore the full combinatorial diversity of all the different cytokine receptor dimers.
In the chat, there are two questions that are the same. What about the immunogenicity related to the various modified cytokines?
Right. Well, that's a good question, and is definitely it's definitely, top of mind when you go into the clinic on these things. But I think we're starting to there are a lot of clinical trials going on right now with different versions of engineered cytokines, and I really can't say anything about our particular cytokines at this time, but it's turning out to not be as big of a problem as one might have imagined. That immunogenicity is not always due to mutations. It's most often due to the behavior of the protein that you're putting into people. And so very well-behaved proteins that have a mutation tend to be pretty well tolerated.
Someone asked, how is ortho IL-2 different from IL-15, if it is, in terms of signaling and overall biological effects?
Yeah, so I didn't get into that, but yeah, the dichotomy between IL-2 and IL-15 is interesting. So, and we've published on this with Warren, they use the same receptors. So IL-15 and IL-2 use IL-2R beta and common gamma chain, and they signal very, very in a very similar way. And so now Ortho-2 differs from IL-15 in that it's completely specific for the orthogonal receptor, whereas IL-15 is specific for the natural receptors, just like IL-2. So IL-15 to me is kind of another way of delivering an IL-2-like signal, but it doesn't solve the specificity issue.
Chris, let me ask you a JAK-related question that I know you've thought about. Not all JAKs are naturally paired with each other in response to cytokines. Like all homodimeric receptors act through JAK2. The common gamma chain use JAK1 and JAK3, but not every pair is physiologically used. And so to what degree, is it that they work better versus that they just weren't combined in the way biology developed?
It's a fascinating question that drives a lot of the way I think about this. I have a slide where I show the missing JAK pairings that nature did not provide for, and I have spent a lot of time thinking about that. One possible interpretation is nature tried those combinations, and for whatever reason they were not good for the host, and they were evolved against. That's possibility. I don't really believe that. We have now created, using cytokines, those non-natural pairings. For instance, there's no JAK2 JAK3 pairing in nature. There's no TYK2 JAK1. There's no and so we've created those pairings artificially using the orthogonal receptor system, and they have interesting signaling properties. And so now we're trying to make ligands to enforce those pairings as well, because I really think there's something happening there that's going to be very interesting.
There are some more questions in the chat. Apologies, I wasn't naming people, sure. But Ross Chiloa asks, I found the ability to re-engineer the cytoplasmic portions of engineered IL receptors exciting. Do you expect that the ability to change the cytoplasmic portion without the need to modify the extracellular portion to be widely applicable? What about other types of immunoreceptors?
Yeah, absolutely. No, I think we're like I said, we want to make the double ortho so we can sample all the dimeric combinations, including the non-natural JAK pairings. And it's been done in other systems as well. I mean, Phil Greenberg was making so-called switch receptors quite a few years ago, where he was making chimeric receptors, for instance, GCSF extracellular domain and interleukin-2 receptor intracellular domain. So the concept's been out there been around a while. What we've done is just make them completely orthogonal. So So I think in cell therapy, this is where the action is right now, is using these type of systems to deliver new signals to cells.
There's a question, Jinhua Liu. Nice data on cancer models. Have you tried engineered IL-2 and other cytokines in HIV?
We have not. We have not. I mean, I'm particularly interested in a new kind of IL-2 we've made that we've brought in a STAT3 signal that we think it might mitigate exhaustion. I'm particularly interested in trying that in virus models where CD8 T cell exhaustion is a problem. So we haven't tried it, but I'm definitely interested.
And there's a question, Danielle Vertelli. Can you expand on the facets of the behavior of the protein that impact on immunogenicity?
Well, it's mainly, aggregated proteins that don't behave well. And I know that doesn't help you much, but for a biochemist, when you say a protein doesn't behave well, you kind of know which way the wind is blowing. And so well-behaved proteins, monodisperse, really happy, well-expressed proteins tend to not be as immunogenic as more aggregated proteins. And it's not a hard science, but that's kind of what the data is showing us.
Okay. I think those are all the questions. Chris, wonderful talk, really terrific, exciting stuff. Thanks very much for once again being at NIH, sort of. Not
well, I missed the reception. I missed the reception afterwards, but we'll hopefully do this again in a couple of years.
Sounds great.
Okay, thank you.
Thanks a lot, and thanks to everyone for the questions.